Start. Welcome back guys. Now in this video let's discuss about the basics of what exactly is a neuron and nerve. Okay, it's like an introduction to the nerve physiology. Okay, so now let's take the heading nerve physiology. Now in this nerve physiology, first we'll be discussing about what exactly is a nerve and neuron. Guys, we all know that from the spinal cord, okay, let's say from the spinal cord, who is going to come out? From the spinal cord, it's the spinal nerves are coming out, okay. So the 31 pairs of spinal nerves are coming out, and from the brainstem 12 pairs of cranial nerves are coming out, okay. Now I am going to take this one nerve, okay, I am going to take this one nerve and I am going to show you what exactly is present inside a nerve, okay. So now I am going to show you the cross section of this nerve. I am going to cut a nerve and I am going to show you, okay, what exactly is present inside this nerve. Guys, here is a cross section of a nerve, let's say. See, inside this nerve, okay, let me make it big so that you will understand it better. Okay, now here inside this nerve we are having a lots of fascicles. Lot of fascicles are present. Okay, these are lots and lots of fascicles are present. Now you will be having it out. So what exactly are these fascicles made up of? Okay, first let me tell you the coverings. This nerve is covered by a connective tissue layer. This nerve is covered by a connective tissue layer, which I am showing here with the violet color. So this covering which I am showing you, this covering is called as epineurium. Okay, this is called as the epineurium. Now, sir, these fascicles, they are also covered by one more layer. Okay, the fascicle is also covered by one more connective tissue layer. Okay, so this covering which I am showing here in green color, this is called as perineurium. Okay, perineurium. Now, what exactly is present inside the fascicle, sir? Okay, see, inside the fascicle you are having multiple axons of the neurons. So here you will be having multiple axons. These are the axons of different sensory neurons or motor neurons. Okay, these are the axons of individual neurons. So inside a nerve, you are having multiple neurons. Okay, multiple neurons are present. There are many neurons. The neurons are coming together and they are totally present inside a nerve. Okay, so the individual functional unit is the neuron and the combination of neurons is going to form this nerve. Okay, so nerve is a part of peripheral nervous system, don't forget. Okay, the cranial nerves and spinal nerves, they are part of the peripheral nervous system. So inside a nerve you are having multiple neurons. Now each neuron, okay, each individual dot is a axon of neuron. They are the axons of individual neurons. Even they are covered. Okay, they are also having this their own covering. So this own covering, okay, in the pink color, so this is called as endoneurium. Okay, endoneurium. So now you understand. So totally how many layers are there? There are 3 coverings. One is the epineurium, perineurium and endoneurium. Now here you will be also having the blood vessels, okay, in between the fascicles. Okay, let me show it with a different color. These are the blood vessels, guys. Okay, even though I am showing with the black color, these are the blood vessels. Now my question here is, see, there is something called as the blood brain barrier and the blood neuron barrier, which means it's protecting. This barrier is actually protecting the neurons from the contents of the blood. In the blood there may be a drug, there may be a toxin, there may be a poison, there may be some, like, some chemical. So the neurons should be protected. So there should be a barrier which is protecting these neurons from the toxin substances in the blood, or maybe bacteria in the blood, or maybe some chemical in the blood. Okay. So who is forming this blood neuron barrier? Blood neuron barrier. So who exactly forms this blood neuron barrier? The blood neuron barrier, is it formed by the epineurium, or is it formed by the perineurium, or is it formed by the endoneurium? What do you think? It is this perineurium, guys. It is the perineurium which is protecting this neurons mainly. Okay, because the cells of the perineurium, see the cells of the perineurium, okay, perineurium is nothing but like a layer, connective tissue layer, and whatever the cells which are present in this perineurium, they are having the tight junctions, okay, in between the cells. Okay, if this is the cell layer, in between the cells, there is going to be very tight junctions. Okay, the gaps are not there. The one cell is very tightly attached with the other cell. The cells here, they are very tightly attached with one another. So it doesn't allow the substances to pass from the blood into the neurons. Okay, so because of this tight junctions, the neurons are well protected. Okay, so now tell me, who exactly is forming the blood neuron barrier? The blood neuron barrier is mainly formed by which layer? Perineurium, the cells along with their tight junctions. And not only that, but also see the blood vessels which are entering, okay, into the nerves, the blood vessel. Let's say this is the blood vessel in the nerve. What are the lining cells of the blood vessels? The lining cells of the blood vessels, these are called as the endothelial cells. Okay, these are the endothelial cells. And these endothelial cells, they are also having tight junctions, okay, which are made up of claudin, occludin. The tight junctions are there. So because of this tight junction, the endothelial cell along with the tight junction and the perineurial cells along with the tight junction, they are forming the blood neuron barrier. Okay, tight junctions are helping in the formation of the blood neuron barrier. Perineurium forming the blood neuron barrier along with the endothelial cells. Okay, the endothelial cell along with the tight junction, and the perineurium, the cells of the perineurium along with the tight junctions forming this blood neuron barrier. Okay, so now let me write here. First, there are nerves. Nerves are covered by layer called as epineurium. This is the summary, guys. Epineurium. Inside the nerve, what do we have? Fascicles. Okay, the fascicles are covered by perineurium. Inside the fascicle, you are having nerve fibers, or you can say neurons. And these neurons are also covered by endoneurium. Out of all, the perineurium is the one which forms, okay, perineurium along with the endothelial cells. Okay, perineurium along with the endothelial cells with the tight junctions. Okay, forms blood neuron barrier. Okay, forming the blood neuron barrier. Okay, now let me go into the details of a neuron. Take the heading structure of a neuron. Structure of neuron. Now let me explain the structure of neuron in an easy way, simple. We know a neuron is going to have a cell body. Let's say here is the cell body of a neuron. Okay, so this cell body is having this dendrites. Okay, cell body is having the dendrites, and there is an axon. Okay, there is going to be an axon and nerve terminals. These are the nerve terminals. Okay, here are the nerve terminals where the neurotransmitter is released into the synapse. Now, what are the important things that you should know? Guys, we are having almost 100 billions of neurons. Okay, we are having hundreds and more than 100 billion neurons are there. In that, okay, where do you see the largest cell body? See, first let's start with the cell body. The cell body is also called as, okay, cell body. It's also called as soma or perikaryon. Cell body, soma, perikaryon, they're all same things. Now, where do we see the largest MCQ? Largest cell bodies are present in which neurons, in which part of the body? Largest cell bodies are present for the motor neurons. Arising where? Motor neurons arising. Arising from anterior horn of spinal cord. Okay, so you tell me where exactly the cell bodies, like the largest cell bodies are present, where the largest cell bodies are present to the neurons which are arising from the anterior horn of the spinal cord. We all know the anterior horns of the spinal cord are motor in nature. The anterior horn is giving rise to the motor neurons. The cell bodies of these motor neurons are the largest in size. And where you see the smallest size cell bodies. Smallest size cell bodies are seen where? The granular cells. Granular cells are also nothing but the neurons in the cerebellum. Granular cells in cerebellum. Okay. So, granular cell is nothing but a neuron. It is having the smallest cell body. Now, within the cell body, we are having something called as the Nissl bodies. Okay. Now, you will be having a question, what exactly are these Nissl bodies, sir? Nissl bodies. So, what exactly are these Nissl bodies? Nissl bodies are nothing but rough endoplasmic reticulum. Rough endoplasmic reticulum with ribosomes. The rough endoplasmic reticulum with the ribosomes is called as the Nissl bodies. See, these Nissl bodies are only present in, they are only present in cell body, not in the axon. Okay. Now, there is a term called as a chromatolysis. Chromatolysis. Now, sir, what exactly does it mean by the chromatolysis? Chromatolysis means is the dissolution of these Nissl bodies. Whenever the neuron is damaged, the Nissl bodies will be undergoing dissolution. So, the dissolution of the Nissl bodies is called as chromatolysis. So, dissolution of Nissl bodies. So, dissolution of the Nissl bodies. Okay. That's it. And where do we see the maximum number of these Nissl bodies? The maximum number of Nissl bodies are present in the motor neurons. Okay. Next, what else we should know? Guys, let's come to the axon part. The axon, okay, is having these coverings. Okay, the axon is having these coverings. So, what are these coverings called as? See, actually it is working like an insulation. Okay, the axon is having this insulation or these coverings. These coverings are also called as the myelinations. Okay, so let me write. There is myelination around the neurons. So, who is going to do this myelination? See, in the central nervous system, okay, in the CNS, in the central nervous system, myelination is done by a cell called as oligodendroglial cells. So, oligodendroglial cells are the cells which are causing the myelination around the neurons in the central nervous system, okay, in the central nervous system. But in the peripheral nervous system, who is going to do the myelination? Myelination is done by Schwann cells. But one exception is there, The exception is, see, actually it's the Schwann cell, which has to do the myelination around the peripheral nervous system. Peripheral nervous system includes the cranial nerves and the spinal nerves. But one exception is one cranial nerve, which is a part of peripheral nervous system is lined by oligodendrocytes. Who is that? Exception. That is optic nerve. Okay, optic nerve is myelinated by oligodendroglial cells. Okay, it's myelinated by the oligodendroglial cells. That's the one important exception. See, in between the myelination, see, here is a myelination, here is a myelinated segment, here will be the myelinated segment, let's say. okay, in between the myelinated segments. Okay, so there are gaps present. So what exactly are these gaps called as? See, these gaps are there. So these gaps are called as nodes of Ranvier. Okay, let me write. So gaps between the myelin sheath. So gaps between the myelin sheath is called as nodes of Ranvier. So what exactly are these nodes of Ranvier? Okay. See, the nodes of Ranvier are the places. The nodes of Ranvier are the places where there is large number of voltage gated sodium channels are present. Okay, large number of voltage gated sodium channels are present which favor the development of the action potentials. Okay, so in the nodes of Ranvier, there is going to be highest concentration of voltage gated sodium channels. The voltage gated sodium channels are going to be more in number. Okay, and where do you see the least concentration? Least concentration of voltage gated sodium channels, the least concentration of voltage gated sodium channels is going to be seen in myelin sheath. Now, let me add you few more important areas. See, the area which I am highlighting here, let us say, this area is called as the axon hillock. Axon hillock, always remember, axon hillock is a part of cell body, it is not a part of axon. Okay. Now, down to the axon hillock, this area which I am highlighting, this is called as the initial segment, which is a part of the axon or the first part of the axon. So, there is a difference. First, this area is called as the axon hillock, and down to it there is initial segment. Okay. So, no, axon hillock means axon hillock is a part of cell body, and initial segment is the first part of axon, is the first part of the axon. Now, where action potential is generated, sir? Okay, when you stimulate a neuron, okay, see, you are coming and you are stimulating here, you are touching here, okay, the neurons are getting stimulated, so I can feel. So, where exactly the action potential is generated? See, the action potential is generated at a different place in sensory neurons as well as the motor neurons. It's a different in sensory as well as the motor neurons. Okay, let me take the heading: action potential generation. Action potential generation. So, where is this action potential generated? In sensory neurons. Action potential is always generated at the nodes of Ranvier. Because you tell me, see, the neuronal ending will be here. Okay, see, cell bodies are not present over here. Cell bodies are present in the dorsal root ganglion. For the sensory neurons, the cell bodies are present in the dorsal root ganglion. Only the peripheral process, the peripheral neuron ending is coming here. So, when I am stimulating here, the nodes of Ranvier will be present. The nodes of Ranvier is going to generate the action potential. The action potential runs along the length of the axon. So, for a sensory neuron, the action potential is generated in nodes of Ranvier, the nearest nodes of Ranvier. But for a motor neuron, the action potential is generated in the initial segment. Okay, this is the best answer. Now, okay, for a motor neuron, the action potential is generated where? In the initial segment. Okay. Now, after this, let me also explain you about the types of neurons. So, how many types of neurons are there? Okay, let me show you. See, here there is a cell body, let us say, here there is a cell body. From the cell body, there is one axon coming, okay, one axon coming, and from this axon, multiple nerve terminals are coming, okay, like this. So, what is this neuron called as? This neuron is called as a unipolar neuron, one cell body with one axon. This is called as unipolar neuron. Next, look at the second type of neuron. Here there is a cell body, and from this cell body, one axon is going like this, okay, and one more is axon is coming like this, okay. So, from here two axons are there, two axons are coming, and there are nerve terminals. like this. So what are these neurons are called as? So these neurons are called as the bipolar neurons. Okay, bipolar neurons. Look at the third type of neuron. There is one cell body, okay, one cell body, and only one axon is coming. Okay, one axon is coming. But this one axon is dividing into, okay, one axon is divided into two processes. One central process and one peripheral process, let's say. So such type of neurons, they are called as pseudo unipolar. Okay, pseudo unipolar neurons, because only one axon is coming out, but it is dividing into two processes, looking as if it's bipolar. No, it's a pseudo unipolar neuron. Okay, pseudo unipolar neuron. Next, look at the fourth type of neuron, which is which are present more in number in our body. That is a cell body with one axon, and see here multiple dendrites are present. Okay, which is having connections with multiple other neurons. Okay, and here nerve terminal, which is also synapsing onto the dendrites of multiple neurons. So such type of neurons are called as a multipolar neurons. Okay, multipolar neurons. So these are the four types of neurons which I want you to know for your exam: unipolar, bipolar, pseudo unipolar, and multipolar. So how the questions are going to come up in your in your exam? Where do you see such unipolar neurons in our body? Actually, unipolar neurons are not present in the humans, but in one place, if you have to pick up one place where the unipolar neurons might be present, the best answer will be mesencephalic nucleus of the trigeminal nerve. So unipolar neurons are present in mesencephalic nucleus of trigeminal nerve. Okay. Next, where do we see these bipolar neurons? Bipolar neurons are seen in retina, olfactory epithelium. Okay. So in our retina and in olfactory epithelium, there are bipolar neurons present. And where do we see this pseudo unipolar type of neurons? Pseudo unipolar neurons are seen in, okay, dorsal root ganglion. The sensory neurons which are present, in the dorsal root ganglia are examples of the pseudo unipolar neurons. And where do we see multipolar neurons? Multipolar neurons are present in multiple places. For example, all the motor neurons. The motor neurons which are arising from the anterior horn of the spinal cord, they are examples of the multipolar neurons. Okay. Now let's continue with the very important topic called as axonal transport. So this topic will again come in your general physiology also, but anyway. So what exactly is this axonal transport, sir? Okay. So the transport along the axons. See, you tell me, see here there is a cell body, let's say. So cell body is the place where the neurotransmitters are synthesized, and those neurotransmitters whatever are produced here in the cell body, the neurotransmitters has to go down into the nerve terminal, yes or no? Okay, the neurotransmitters whatever are produced over here, those neurotransmitter has to be transported to the nerve terminal so that they can be released into the synapse, leading to muscle contraction, or like it's going to produce the action potential to the surrounding neurons. So how this axonal transport is going to happen? Okay, so there are certain proteins called as molecular motors. Okay, there are proteins called as molecular motors. So what exactly are these molecular motors? Let me tell you. See, inside our cells, okay, inside this neuron, let's say, there is something called as a skeletal system. Okay, there is cytoskeleton inside the neuron. For example, I am just enlarging this area. Okay, I am just showing this axon. Now inside this axon, there is a cytoskeleton, let's say, a cytoskeletal filament called as microtubule. Okay, microtubule. This microtubules, a microtubule is made up of what? Tubulin. A protein called as a tubulin. This microtubule, they are having positive end and they are also having a negative end. The positive end and negative end. Positive end means the end which will grow, always grow. With the addition of a new tubulin molecules, the positive end is always going to grow, and negative end is not, okay, let me put it this way. There's going to be a positive end, okay, there's going to be a positive end and there's going to be a negative end. The positive end means the end which will always grow, and the negative end means the end which will not grow. Now, this microtubule, always remember, it is going to act like a road, guys. It is going to act like a highway. Now, on this highway, actually, these molecular motors will actually walk. They are actually walking. Just like cars going on a highway, cars which are going on a highway, these molecular motors actually walk. They will actually go on this cytoskeleton. So, what are those molecules called as? Let me say here, look at this molecule. This molecule, okay, it is actually moving towards the positive end. And there is one more molecule, okay, there is one more molecule which is going to walk towards the negative end. These molecules are actually transporting the substances within the neuron. So, these substances, okay, which I am highlighting here, these substances, they are called as the molecular motors, okay, helping the transport of the vesicles or the neurotransmitters. Now, the protein which is going to move, okay, look at here, this protein which is going to move towards the positive end, the transport is called as the anterograde transport. Anterograde transport. And the opposite movement means the protein which is moving towards the negative end, that is called as a retrograde transport. So, anterograde transport and retrograde transport. So, in your exam, they will be asking you two important MCQs. So, what is the name of the protein which is helping in the anterograde transport, and what is the protein that is helping in the retrograde transport? Okay, the movement of the substances. Anterograde transport is with the help of a protein called as a kinesin. And retrograde transport is with the help of a protein called as a dynein. Okay, kinesin and dynein. Now, what else they will ask you is, what is the speed of this kinesin? This kinesin actually will walk and will help in the transport of the substances with how much velocity? What is the speed of this kinesin? The speed of the kinesin is 400 millimeters per day. Okay, and the speed of the dynein is 200 millimeters per day. So, this is how the axonal transport is going to happen with the help of the two important molecular motors called as a dynein and kinesin. How you can remember it for your exam? Always remember, you guys are doctors, dynein for helping in the retrograde transport. Dynein helps in retrograde transport. What is the clinical importance for this is, see, there is a virus called as the rabies virus. Okay, rabies virus and tetanus toxin. Okay, rabies virus and tetanus toxin. What do? See, they are going to, okay, see, whenever this whenever the rabbit dog comes and bites you, the rabies virus is going to first enter into your body. Now, this rabies virus, okay, it will enter into your neurons. It's going to first enter into your sensory neuron. After entering into your neuron, it goes and binds with the dynein. So, what the dynein will do? Dynein is going to move, this dynein is going to move the virus into the central nervous system. So, how the rabies virus and tetanus toxin is going to enter into your central nervous system? By using this dynein. So, this rabies virus and tetanus toxin, they use dynein to enter into central nervous system. Okay, central nervous system. So, this topic is very, very important for your exam. With this, what we have completed? We have also completed the axonal transport. So, in the basic, see, this is going to be a basic introductory video where we have discussed about what exactly is a nerve and a neuron. Again, I am concluding what we have discussed. A nerve is a combination of multiple neurons. In a nerve, you are having multiple neurons are there. There might be a sensory neuron, there might be a motor neuron. Multiple neurons are present within a nerve. A nerve is having a connective tissue that is called as a epineurium. Inside it there is perineurium, and inside it there is endoneurium. Okay. Tell me, who is forming the blood neuron barrier? The blood neuron barrier is formed by the perineurium along with its tight junctions. The endothelial cells along with its tight junctions. Okay. Now, where exactly the voltage gated sodium channels are present. The voltage gated sodium channels are rich, high in concentration in the nodes of Ranvier. Who is going to form the myelination? Myelination is formed by oligodendrocyte cells in the central nervous system and Schwann cells in the peripheral nervous system. What exactly are the micelle bodies? The micelle bodies are nothing but the rough endoplasmic reticulum along with the ribosomes. Okay, and the dissolution of this micelle bodies is called as a chromatolysis. That is going to be seen in the damage to the neurons. Okay. And what exactly is the difference between the axon hillock and the initial segment? The axon hillock is a part of the cell body and initial segment is a part of the axon, first part of the axon. Where the action potentials are generated in a sensory neuron, action potential is generated in the nodes of Ranvier, but in the motor neuron, action potential is always generated in the initial segment. How many types of neurons are there? 4 types of neurons: pseudo-unipolar, multipolar, bipolar and unipolar. Unipolar are present in the mesencephalic nucleus, bipolar are present in the retina, pseudo-unipolar are present in the dorsal root ganglion, multipolar neurons are present in the meriplaces. Example is a motor neuron. Okay. So with this, I am concluding the topic of nerve and neuron. Hope the video is helpful. See you in the next video. Thank you.